Hi, everyone, and thank you, the organizers, for this kind invitation. And I'm going to talk about an understudied and underappreciated side of aging, which is the reproductive aging. So just to start with one disclosure, some of the stuff that you're going to see here is being commercialized, and we are developing in the spin-off company started from my lab for Eve. We are developing small molecules to extend aging of oocytes in the IVF clinic. So my business is the oocyte and female reproductive aging. And oocytes develop from primordial germ cells, which develop and go into meiosis. And all this is happens in the embryo during embryonic development. And as the embryo grows, all the oocytes arrest and do not continue meiosis from this point onwards until they reach puberty. And with puberty, the oocytes start to grow in groups, and until one in human or several in other mammalians are selected and reach the complete ovulation. So we are very, very interested in this huge pool of arrested oocytes, which remain in this form in most of the life of the female. And what I'm going to tell you today is that, at least from the epigenetic angle, these oocytes are very unstable. And we need to understand the processes that they undergo in order to treat infertility. Okay, so the first story I'm going to tell you has been published last year in Aging Cell, and this is about the aging of the oocytes. And we know that female reproductive aging is actually it's the fault of the oocyte. The oocyte is actually the main culprit in this reproductive aging. If we look at live birth rates in IVF with the age of the mother, we can see a very, very sharp decline, but we can completely stop this decline and restore the original percentage if we use a donor egg at the age of 25. So it's not the endometrium, it's not the successive embryonic development, it's actually all about oocyte quality. So in order to start to study the fertility and the oocyte quality and how this is affected with the age, we wanted to put aside one factor which we know has been studied very extensively in the last few decades, and this is that with age, the oocytes become aneuploid. And we wanted to see if there is something more to this oocyte quality than just the aneuploidy. And for this purpose, we made use in mice of an age, and here it's written old. So this is the age of 9 months old, and we can see that at 9 months old, the oocytes are still euploid. There is the correct number of chromosomes. However, when we look at the maturation rates in the dish as one of the measures for fertility, and there are multiple measures, we can see that the level is already reduced at this age by half. So this gives us a tool, an opportunity to see the other factors besides aneuploidy that contribute to this oocyte age. So what we started to do was to look at the epigenome as one of the things, and the talk before me also mentioned that the epigenetic changes are one of the hallmarks of aging. So we wanted to look at that. And we stained for several epigenetic markers, a lot of oocytes. And what I'm showing you here is two markers that are representative of the repressive part of the epigenome, which we call heterochromatin. And you can see here for this marker, H3K9 methylation, which is a histone modification in heterochromatin. We can see the loss of signal from the young group to the old group, and this is true both for H3K9 methylation and H3K27 methylation, another marker. A very important control is to look at the active part of the genome, an activating mark, and in the fraction of the epigenome that we determine as euchromatin, which is the activated part of the epigenome where most of the genes lie. And we wanted to see this for two purposes. First, to show that we do not lose the entire nucleosome with age, and also technically that we can stain older oocytes as well as we can stain young oocytes. And you can see that there's no significant difference between the two groups for this marker and other markers that we check. So actually, the loss of signal, of epigenetic signal, is specific to the repressive part, to the heterochromatin. We asked whether this loss of heterochromatin continues to be very significant when the oocytes progress into ovulation, and these oocytes are in the middle of the process of ovulation, and actually they are during the first meiotic division. And what we did here was to do a chromosome spread of the chromosomes inside the oocyte, and you can see that we stain these chromosomes for the same marker, H3K9 methylation, and actually the difference is even bigger. So this loss continues until the ovulation of the oocyte and even get worse. And it's not because we take out the oocytes from the ovarian environment that they lose the heterochromatin. And we can look at the oocytes while they are embedded within the ovary. And these are cryo-IF sections. And you can see what the situation here is in the young, and the green signal is the H3K9 methylation, and what happens in the oocyte in the older group, and you lose most of the signal. And we did not look only in mice, we also looked at human oocytes. And you can see here human oocytes, and we got human oocytes from the clinic at the GV arrested stage. These are oocytes that are not needed by the patients, to about 25% of the oocytes in IVF. And we asked for permission, and we got the oocytes for research purposes, and I'm very grateful to all these patients who donated these oocytes. And you can see under 35 years old, we get a very strong H3K9 methylation signal, and below we get much less. And you can even plot the dots when each color represents another patient, and you can see that it's kind of a biological clock of the intensity of H3K9 methylation with age in the oocytes. So now back to mice, and the question of, okay, we lose the repressive part of the genome, what the consequences are. And we looked at something which consisted about half of our genome, and this is transposable elements. These are small virus-like particles that can jump from place to place inside our genome. And the most damaging of these elements is the line elements. And here we stain for one of these proteins that the line elements make, the L1OF1P, and you can see that there's a big elevation from the young group to the old group, again, back to mice. And my student Pera did also a really heroic experiment of Western blot to show the same thing. There's about 300 oocytes per lane here, all picked by hand. And also when we did small RNA-seq on the two groups of young and older oocytes, you can see that it's not only the retrotransposon, not only the lines that are overexpressed, but actually all the repeats in the genome are overexpressed. These are the places in the genome that were before repressed by this fraction of heterochromatin, and now in the older group they are activated. So what is this activation of retrotransposons doing? So part of it is that the retrotransposon can make copies of themselves, reinsert in the genome, and as part of this process they create DNA damage. And you look at this marker for DNA damage, gamma H2AX, and there is a significant rise with age in the old group. And when we looked at RAD51, which is a marker of DNA repair, you can see that it goes into the nucleus of the oocyte. These beautiful images show that it happens only in the older group and much less in the younger group. Okay, so what I've shown you actually is a correlation only between the loss of heterochromatin, the activation of retrotransposons, and the DNA damage. And what we wanted to do was to actually correlate these things in a causal manner. So we use different drugs in the dish with the oocytes. The first drug that we use is called Pitocin. Pitocin is a very called 39H1 and 2. And when we use Pitocin in the dish, you can see that we, as a part of it, this inhibition, we lose most of the signal of H3K9 methylation. As a result of that, we activate in these oocytes retrotransposon expression, showing the link between these two things. And as a result, we mature the oocytes much less proficiently in with the Pitocin, showing that the maturation ability of the oocyte is linked to the level of the heterochromatin. Can we do the opposite? Can we actually intervene in all the oocytes and get a better result? And the answer is yes. So we did it on several levels. The first level is the intervention at the level of the retrotransposon. So when we used a reverse transcriptase inhibitor called AZT, this blocks the entry of the retrotransposons back into the genome. What we get is much less DNA damage in all the oocytes and a better maturation rate in vitro. But it's not the only level where we can intervene. We can actually also intervene at the level of the epigenome. This molecule is called SRT1720, is an activator of a very important heterochromatin factor called sirtuin1. And when we use this molecule, we elevate the level of H3K9 methylation and we lower the level of the L1 retrotransposon. And a similar effect can be seen when we put a plasmid inside all the oocyte, overexpressing this factor, which is also an important factor of the heterochromatin. It's the enzymes that put this modification, H3K27 methylation. And when we do that, we get much more H3K27 methylation. We get less expression of the retrotransposons and an elevated maturation in vitro of the oocytes. So all these things eventually can serve as therapy. So just to summarize this part of the model, and with age, we see that the chromatin loses the heterochromatic marks. As a result of that, a lot of places in the genome start expressing. Part of them are retrotransposons that eventually go into the genome, causing DNA damage. And these oocytes do not mature and cannot complete the IVF process. And right now, what we are doing is we are in the middle of IVF experiments for mice with these molecules that we described to show that we can actually make older females have better chances in IVF with this treatment. Okay, so that was the first part. The second part is about what happens to prepubertal oocytes and what happens epigenetically during pubertal transition, which is also very important. So what I'm going to show you here is a figure from Eva Hoffman's excellent paper showing that it's both ends of the spectrum of the age that have a fertility problem, and oocytes in both ends have problems. And we wanted to characterize this end as a mirror to the aging problem. This is not only a theoretical problem because of cancer patients and cancer survivors. So these cancer patients, when they are pre-pubertal, before puberty, and they are going to go to undergo a gonadotoxic treatment, and then one of the only options for treatment is taking out the ovaries and cutting them to pieces in the dish, and then when hopefully the patient is in remission, putting the piece of ovary back into the body and doing IVF. In some of the patients, this is not possible. For example, in leukemia patients, because you don't want to introduce back again blood cells, which might be cancerous. And for this purpose, we need to be able to take out oocytes from these ovaries and mature them in vitro. And this is the purpose of this project that I'm describing. So we started to characterize the epigenome of these oocytes, and like in aging oocytes, also prepubertal oocytes have much lower heterochromatin than the mature oocytes. You can see this in these images here. And also they have a higher level of retrotransposon expression at the level of RNA than postpubertal oocytes. However, the oocytes somehow manage to keep the protein expression of L1 retrotransposons in check, and you can see that there's not more expression of the retrotransposon protein. And we think that this might be because of the immune system of the oocyte. One of the most important proteins in this immune system is the disoprotein, and you can see that this is much elevated in the prepubertal oocyte compared to the mature, providing the prepubertal oocytes with a tool to counteract the expression of retrotransposons at this stage. Like we did for the aging oocytes, we also looked at human oocytes, and we can see the same thing as we see in mice, prepubertal oocyte compared to mature oocytes, and we have an elevation of H3K9 signal from one of the groups to the second group, and showing that this is conserved. So we tried the small molecules that I described before also on these prepubertal oocytes, and unfortunately these do not work. They do not work at all, and we are still not sure why. There might be an active mechanism on the prepubertal chromosome which stops the activity of the molecules I described for the age. But in order to try and still mature better the oocytes from the prepubertal fraction, what we did was to use hormones in the dish and see if the hormones have an effect, like FSH, for example, on the status of the oocyte. And when we use FSH in the oocyte in vitro, one of the things that can happen is that we can elevate the level of H3K9 methylation that I showed you before. And at least from the epigenetic point of view, the oocyte will reach a better point in maturation. But when we look at the actual maturation and the separation of chromosomes, we can see that while in post-pubertal oocyte in mice, we get full separation of the homologs in the dish, and in the prepubertal, we get almost nothing. And the FSH treatment is very limited. Okay, so just to conclude, epigenetic damage occurs during oocyte aging, and epigenetic remodeling events occur with puberty. The damage is treatable and reversible in vitro, and we are working for the aging oocytes, and we are working to bring this to the clinic. And the drug that we will use will be called Epicova. The remodeling event in prepubertal oocytes can be partially mimicked in vitro, and we are working to make this efficient and better, and to establish what we call IV-puter, the in vitro pubertal transition. With that, I will finish. I will thank my student Peira, who did all this work, and my technician Eli Reich, and help from both Shaare Zedek and Hadassah hospitals, where we had the clinical collaboration. So thank you for your attention. And if there are any questions, I will be happy to answer.